Hey, it's Mr. Donahue again. This time we're going to take a look at 25.8 and 9 stuff. Biochemistry Basics. We're going to describe the structure of amino acids, polypeptides, and proteins. So what is a polymer? Well, basically, poly means many, right? And mer means parts, so it's made up of many parts. So many monomers, which means it's like one kind of thing, are linked together to make a much bigger molecule. So here's my example. We got 4 different monomers that end up linked together to give us one big molecule. So this process is known as polymerization because you're making a polymer. So polymerization. So biopolymers. Bio has to do with living things. So obviously we're going to be talking about polymers found in living systems. So proteins, such like amylase, the digestive enzyme in saliva, right? So its monomer is going to be amino acids. So proteins are made of amino acids. We're going to talk about polysaccharides, where their monomers are simple sugars. So it's about making a polymer from sugars. And nucleic acids, which are made up of those guanine, cytosine, adenine, thymine, and uracil if you're talking about RNA. And these are the things responsible for our genetic code, our DNA molecules, our chromosomes. Cool. So proteins. Proteins are polymers that are of critical importance to biological systems, right? They are the enzymes. Enzymes are proteins that protein catalysts that carry out reactions in cells. You want photosynthesis to occur? You need an enzyme. You want cellular respiration to occur? You need an enzyme. You want to break down that food so that you can use it? You need an enzyme. So proteins are super important. So they're made of amino acids bonded together. And this is an amino acid, a generic amino acid. Why are they called amino acids? Well, let's think about this. Amine. Oh, what do you think? There's an amine group? Hey, would you look at that? You got an amine group. And what about an acid? Oh, hey, look over there. You got an acid group. So that is where they get amino acid from. You have an amine group and a carboxylic acid group. So proteins. These R groups on the amino acids can vary. So this is the genetic skeleton of skeleton of an amino acid. You have this amino group, you got a carbon, and then you got a carboxylic acid group. And on that central carbon, there is a hydrogen and then something else. And this R group determines what kind of amino acid it is and its properties. So different R groups have different properties. For example, it could be something as simple as another H, or it could be something as complex as this one right here. And because they have different substituent groups, because these parts are different, they have different properties. So proteins, again, amino acids, polymerization occurs and peptide bonds are formed. So polypeptide is formed when many amino acids are bonded together. And how do they bond together? Well, a condensation reaction. So you take the OH from the carboxylic acid end and an H from the amino end, and you remove water molecule. So you subtract water, you make these two things now come together and bond together by the remaining nitrogen that's still left there. So you end up with something that looks like this. So you got your carbon here. What used to be the acid group, the OH group, has been removed, and now it's bonded at that amine group to the other amino acid. And that is what we call a peptide bond. All right, so proteins, their primary structure is the sequence of amino acids. So proteins are really complex, and we have different ways to describe their structure. Primary structure is just the sequence of amino acids. So you got these are your amino acids: go, phi, lu, ser, and cis, and those are just a sequence. It's what is the order of the letters. The secondary structure is, all right, now that I have this chain of amino acids, what happens to it? Well, the amino acids are going to fold onto themselves and they can form smaller structures. And we have ways to describe those smaller structures. So you can see over here, we got an arrow, which is showing one kind of structure, and we got this helix, which is showing another kind. And the two structures we're going to take a look at are beta pleated sheets and alpha helixes. So alpha helixes. Basically, the polypeptide twists and folds on itself in such a way that the NH group donates a hydrogen bond to the C double bond O group of another amino acid, and that ends up making a helix shape. So if you take a look, here you have your nitrogen with the hydrogen on it, and it's donating a hydrogen bond to the oxygen on that carbon-oxygen double bond. So it's able to interact with itself. It's not even interacting with other proteins, it's interacting with itself, twisting to give you that double helix, that spiral shape, right? Another possible shape is the beta pleated sheet. So it just looks like a pleated sheet. So the polypeptide chains line up laterally, forming a twisted sheet. So we have our chain going this way, and then it can twist around and go this way, and it's interacting with itself that way. And you can see again it has to do with hydrogen bonding. So the nitrogen donates a hydrogen bond to that double bond O on another part of the chain. All right, so we have tertiary structure, which is all right, we have these beta sheets and these alpha helixes, but overall, what happens to the shape of the molecule? So it's the overall shape of the protein. Is it going to be a globular protein where it looks like a glob? Or is it going to be a fibrous protein which looks like a fiber? It's longer than it is like wide. It looks like a fiber as opposed to a glob. So this is determined by its sequence of amino acids, its folding, and its interactions with its surroundings. So say, for example, it was surrounded by a polar solution. All those non-polar amino acids are going to bunch up together in the center of the molecule so that it's, away from the water because they're hydrophobic. They're non-polar. pH can also protonate or deprotonate some of the amino acid side chains, which can change its charge. And if you change its charge, you change its behavior, and it'll change how the protein folds in its shape, which is why you always see like pH and then enzyme activity, and there's usually like a peak somewhere at an optimal pH. That's how pH affects enzyme activity. And then we have another one. So primary is the sequence. Primary structure is the sequence. Secondary is the folding. Do you get alpha helixes or beta sheets? Tertiary is the overall shape. Is it globular or fibrous? And then we have this thing called quaternary structure, which I'm only going to mention briefly. It's basically a complex of protein molecules. So it's not just one molecule anymore. You've taken multiple protein molecules and had them interact to make one functional unit. So for example, hemoglobin is really made of 4 protein subunits that come together to carry out a single function, which is to carry the oxygen in the blood throughout your body. So even though this is made of 4 different chains, it has one function, and that would be its quaternary structure. So shape and function. Why is shape so important? The shape of the protein determines its function. If you change its shape, you change its sequence, it's not going to do the same job. So take a look at this. You can see, hey, it kind of fits there. And what's really interesting is if this is my substrate and it binds to my active site, it can change the active site. And we call this an induced fit model because once it bonds, then that's when it fits perfectly. And then it can do its activity. So it's like a lock and key model. All right, so what can you what can you tell me about proteins and their structure? And that's what that whole video was about. So hope you found it helpful. See you in class.